Hello, this is Dr. Anderson, and we are going to continue the journey in sensory physiology by talking about the pathways that connect the periphery and the central nervous system. So we want to think about some anatomy, how are action potentials entering and exiting the spinal cord, how is the body organized in terms of sensory information, and essentially what that means is what are dermatomes. We need to understand the anatomy of the dorsal column pathway versus the spinothalamic column. These pathways are different in their anatomy, they're different in their speed and their discrimination, they're different in the kinds of modalities they carry. And then we're going to do a couple of application problems. One is with stereognosis, and the other is with something called the Brown-Séquard syndrome. So neurons are classified in terms of fiber size. A fibers are the largest, and they're myelinated. So mechanoreceptors, motor neurons, proprioception, fast pain are all using those large fibers. B fibers are, this should say fiber and not filter. Those fibers are medium size and they're myelinated, and this is what we're going to see in the autonomic nervous system. And then finally, C fibers are small and unmyelinated. So they're the slowest. That's the slow pain, the itch, and the temperature. So how do action potentials get into your system? Well, we're going to have a first order neuron that comes from the skin and the muscles, and it's going to enter the spinal cord and either synapse right away in the spinal cord or project up to the medulla and then synapse. Then there will be a second neuron that crosses the midline and gives us something called contralateral representation. In other words, sensory information from the left side of your body goes to your right brain. And those action potentials are transmitted to the thalamus, where there's another synapse. Then there's a third order neuron that arises from the synapse and goes to the parietal lobe, where we find the primary somatic sensory cortex, and that's going to tell us what we're sensing. So your body's very organized. We have bilateral symmetry, and we have 31 pairs of spinal nerves. So early in fetal development, actually embryonic development, we're not even a fetus yet when this stuff happens, you get a formation of spinal cord and these 31 little nodules called somites. So you have a vertebra, you have two spinal nerves coming out of each vertebra, and they innervate a layer of the body. So we have C2. It's hard to see C1 because not everybody's got a C1, and C1 is deep. C2, 3, 4, and 5 innervate the diaphragm. Remember, 3, 4, 5 keeps you alive. Then we have our thoracic dermatomes, lumbar dermatomes, and the sacral dermatomes. They're a little different on the front and the back, and they're not all the same for all the modalities. So the touch dermatomes may not be exactly like the pain dermatomes, but this organization into dermatomes is the idea that I want to get across here. So we look at our sensory tracts. Here are both of them here from your textbook Vander. Notice the spinal cord. There's a butterfly of gray matter in the middle, and then the surrounding is white matter. So gray matter are the cell bodies and synapses. The white matter is the myelinated axons around. Sensory information comes in the back. There's the dorsal root ganglion. That's the cell body of those sensory neurons. Enters the dorsal horn and may synapse right away or may rise in the dorsal columns and synapse somewhere in the brainstem. So we have two pathways. The anterior lateral system, also called spinothalamic. So it goes from the spinal cord to the thalamus is how it's named. And deals with anterior or lateral spinal tracts. So here's our sensory neuron. There's the cell body. Notice it synapses right away in the dorsal horn, crosses. Now we have contralateral representation. It rises on the contralateral side of the spinal cord. There's some collaterals that go off to, for example, your reticular formation. It synapses in the thalamus. Then a third order neuron goes to the cortex, so you know what you just felt. So this is a lot of pain and temperature, some crude sensations, crude touch, itching, those kinds of things are in this pathway. All right, the dorsal columns, there's our dorsal root ganglion. The neuron comes in and does not synapse right away. It rises in the dorsal columns on the ipsilateral side of the body, synapses in the brainstem. We have collaterals that go to the reticular formation and some other areas. Rises to the thalamus. Third order neuron goes to the cortex so we can see what it is. Now this is a picture from an anatomy, like an A&P textbook, so we can get a little more detail. So this shows us the dorsal columns or posterior column medial lemniscus pathway. This is a discriminating pathway. This is a fast pathway. We have more myelinated neurons, and we have very nicely mapped dermatomes. We can localize this very well. So, discriminating touch, stereognosis, which is being able to figure out what an object is by touching it and not looking at it, proprioception, weight discrimination, vibration. So, this is a discriminating pathway. So the sensory neuron enters the spinal root. There's the dorsal root ganglion, crosses through the gray matter, does not synapse, rises in the dorsal columns, and finds a nucleus in the medulla. The outer nuclei, that's the fasciculus gracilis. That's coming from the lower body. So I think about a graceful ballerina pointing her toe. The bottom of the body rises in the fasciculus gracilis. It is going to synapse in the nucleus gracilis. The second order neuron is going to cross, giving us contralateral representation, project through the medial lemniscus, synapse in the thalamus. The third order neuron goes to the primary somatosensory cortex, so you know what it is. If I was talking about the thoracic dermatomes, so the torso and the upper body, those are going through the fasciculus cuneatus. So that would come in and rise in the fasciculus cuneatus. And then rise on the ipsilateral part of the body, synapse in the nucleus cuneatus, cross, we get contralateral representation. We go through the medial lemniscus, synapse in the thalamus, and project to the brain. So, I wonder if I can write on this thing so you can see what I'm thinking here. Let's see if I can make this pen work. And I'll write in red. Okay. So, I want you to think of building the body from the bottom, well, bottom up. So at the bottom is your toe, and that's going to be on the inside in the fasciculus gracilis. That's the ballerina pointing her toe. Then projecting to the nucleus gracilis, crossing, and then going to the thalamus. Then the upper body is built on the outside in the fasciculus cuneatus, so this could be coming from a dorsal thermatome or the arms, going in the fasciculus cuneatus up to the gracilis cuneatus, crossing and giving us contralateral representation. So the toe is on the inside, then the hands would be on the outside. So the ballerina with her pointing her foot and the body building up from the bottom up, that's how we remember what's fasciculus gracilis and fasciculus cuneatus. Maybe you could practice drawing this. The nurse anesthesia students have to know this for their boards, so we all have to know this for the next exam. And we can work on it and talk about it in class too. Alrighty. Okay, so what is stereognosis? Stereognosis is being able to pick up a object and figure out what it is. So I'm sitting in my office, I'm going to reach over to my little pencil tray and I'm going to pull something out, and I know it's a writing implement, and it's got a little clip on the side, and I feel the end, and it's got a little eraser. If I push on it, I see a little lead coming out. So this is a mechanical pencil. I'm not looking at it, but I can tell from manipulating it in my hands what it is. That's stereognosis. So what would a stereognosis test say about your parietal lobe? If you had, say, an elderly patient and you gave them a coin or a pencil or a key or a spoon or a pair of scissors and they couldn't figure out what it is, that would say they have some degeneration in their parietal lobe because they can't identify they can't identify the object. And so we would predict somebody with Alzheimer's, and particularly as that progresses, they might not be able to tell what objects are just by feeling them. Alrighty. So here's the other pathway: anterior lateral spinothalamic. That's a less discriminating pathway. The dermatomes are not as organized. And we have a lateral pathway and a spinothalamic pathway. So there's our receptors coming in from the body. There's the dorsal root ganglion. Synapses right away. Right away. It's going to synapse in the gray matter, cross. Some of them will be anterior, some of them will be lateral. They will rise on the contralateral side of the spinal cord, synapse in the thalamus, project to the somatosensory cortex, and then we'll know what it is. Okay, so synapses right away in the spinal cord, crosses in the spinal cord, rises contralaterally, synapses in the thalamus, third order neuron to the cortex. All right, now it's not like you're going to see Brown-Sequard syndrome every day, but it's an interesting problem. Hemisection of the spinal cord. So somebody's got their spinal cord and there's been a hemisection where actually one side is blocked, but the other sides can still show information up to the brain. Okay, so what would be the deficits of a left hemisection? So if this is the point of view of our patient, this is their left side. You say, oh, but it's on the right. No, 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 no. It's their perspective, so it's the patient's left. And a lower thoracic dermatome, the right side is intact. What would be their deficits? Well, this is a way to work it out. So there's a hemisection on the left side. That means things below it are not going to be able to go through. That's the contralateral spinothalamic and the ipsilateral dorsal columns. You see, if we cut right here and we feed back, so temperature and pain on the opposite side, discriminating touch on the same side. The spinothalamic tract below the lesion, below that, has already crossed and is rising contralaterally, and then the contralateral, so the ipsilateral is already crossed of the spinothalamic, and the dorsal columns have not crossed yet. So they will lose contralateral spinothalamic and ipsilateral dorsal columns, and of course they'll also lose motor going to the left side of the body. So draw yourself a diagram like that and you'll be able to figure it out, and I can show you that in class as well. So contralateral spinothalamic, ipsilateral dorsal column, ipsilateral motor function, because that's already crossed. Okay, so then we got to get the information up to the sensory cortex. So right here is the somatosensory cortex, and we have an interesting map in the somatosensory cortex. It's called somatotopic representation. There's a map of you. There's a little map of you right in your somatosensory cortex. Notice there's some association cortex that helps you make sense out of what you're touching. There's the visual cortex back here. Right in front is the motor cortex. So we'll talk about some of those things in class and when we get done with sensory. So again, here you see motor in red, here motor in red, and then somatosensory cortex in blue. Alrighty? Now, here's the little map of you. And notice you are upside down. So the bottom part of your body starts at the top and then goes around the side of the cortex. Now notice the map isn't even. So this map is called a homunculus. It's a person inside of a person. Notice the whole body is in this part of the cortex. Look how much is devoted to the hands. Well, and the fingertip. Look at that. Well, what's going on with the fingertip? Huge two-point discrimination. Also notice the sensation in the face, and notice on the lips. The lips are very, very discriminating. If you've got a 2-year-old, you see them investigating the world with their mouth, of course. There's some sensation of internal organs. What's going on with the genitalia? Aren't the genitalia very sensitive? They are, but they're not very discriminating. So you don't need a lot of cortex. It's about, it's about intensity. It's not about discrimination. So sensitivity and intensity are different than discrimination. I want you to think about that, and if that's not making sense, we need to discuss that in class. So the interpretation happens in your brain. Your hands don't feel, your brain does. Your ears don't hear, your brain does. So how does your brain know where something's coming from? Well, it has to do with the labeled line. There's a spot in your periphery that travels along a labeled line and goes to your brain. And when that shows up in your brain says, oh, that came from my left forefinger. How does your brain know about intensity? How many action potentials did you send? If something is more intense, a higher frequency of action potentials will go to the brain. How are you going to know what kind of stimulus? There are pathways. There are specific labeled lines devoted to touch. There are labeled lines devoted to cold. There are labeled lines devoted to pain. So again, you know where it is and you know what it is based on the pathway it traveled. And resolution, 1 point or 2. Well, there's two aspects to this. We've talked about two-point discrimination out in the periphery, that you have lots of receptors with small receptive fields, so you have a lot of pixels in your camera. But also, all of those receptors are connected to a lot of cortex in the brain. So your hands and your lips have a lot of cortex devoted to it because we have a lot of sensory receptors capable of great two-point discrimination. All right. Another thing that happens in the central nervous system to process information is called lateral inhibition. This does not happen in the periphery. This does not happen at a receptor level. You have to get into the central nervous system where there are synapses to see the effects of lateral inhibition. So there's a stimulus out here. So we've got three neurons and three receptors. So A, B, and C. And the stimulus is coming in to B. There's a little bit of the stimulus coming to C and A. So we have some frequency of action potentials. So we have 4 action potentials here, and in the middle, 2, 4, 6, 8, 10, 12, 14, 16, 18, 19, 20, something about that. So let's say there's 20. All right. So we have action potentials coming up through this sensory neuron. We have a smaller frequency coming on the side neurons. These neurons have collaterals that synapse on inhibitory interneurons. So as the stimulation comes through the neuron, these all stimulate collaterals which synapse on the axon. So this is an axo-axonal synapse. So this is a presynaptic inhibition. And that reduces the neurotransmitter release at this neuron and reduces the neurotransmitter release at this neuron, so that in the second order neuron we only have 1 action potential. So we went from 4 to 1. And here again we went from 4 to 1. In the middle, this one's also being inhibited. But this inhibition is less because the original stimulation on the lateral ones is less as well. So we started out with 20, and now we have, boy goodness, my poor little eyes, 2, 4, 6, 8, 10, 12, 14, 16 maybe, okay? So we had 20 to 4, or 5 to 1. In the central nervous system, we have like 16 to 1. So this has fewer action potentials than this, but the ratio is greater. So what we've done is intensified the center and inhibited the sides. What does lateral inhibition do for us? It helps us specifically localize things. If there's a little tiny overlap in our neurons, that really helps us hone in on the location of something. It also helps us with edge detection. Edges. That's something we really want to know. On my computer here as I'm recording, I want to know where the edge of the computer is. I want to know where the edges of things are. There's a lot of lateral inhibition in the visual system because you want to know where one object ends and the other object starts. Edges are very important pieces of information. So lateral inhibition is very important for that. So here's another picture from your textbook. Here's the area of receptor activation. You've activated all 3 of these neurons, but there's most activation in the middle. So they all inhibit each other, but the central one is going to inhibit the sides the most, so that we intensify the middle and we improve localization and improve edge detection. So we need to talk about the difference between sensation and perception, okay? When you send action potentials to your primary cortex, primary sensory cortex, it tells you sort of when and what and how intense. So what is the stimulus? It's a touch. Where did it come from? It came from my index finger. What's the intensity? It's about a 2 on a scale of 10, or whatever it is. Those are bare bones kind of information. But we actually can get a lot more information from texture and complexity and what the stimulus means. Oh, that's a very smooth, round, raised thing. That's a button. That's the elevator button. I need to push that so I can bring the elevator here. I mean, it's there's a context for what it is you're feeling. So a lot of people use sensation and perception interchangeably. I'd like you to not do it that way. Sensation is just, can you detect the stimulus? Can you detect it? Can you tell what it is? Can you tell where it's coming from? Can you tell how intense it is? Perception is, what's the context? What does this mean? Have I heard this before? Have I seen it before? Have I felt it before? What does it mean? What do I do when I feel this? The difference between sensation and perception is also, look at this slide you're looking at. You can see a bunch of black and white stuff, right? There's a bunch of black things on a white screen. But you can see that, this one is bolder than that, but it's so much more than that. These are words, and you can read them, and you know what they mean. Your simple primary sensory cortex cannot do that for you. So your primary cortex has to communicate with other parts of your brain to make sense, to provide context for what it is you're sensing. All right, so that concludes our second presentation. As you can see here, after this, we're going to talk about pain. So think about what your questions are, think about what you want to talk about in class, and I'll see you next time.